Hello everyone. Welcome to this Shimadzu sponsored webinar. I'm Nazim Boutagu, Life Science Product Manager at Shimadzu Scientific Instruments. I will be your moderator today, and the topic of discussion is OmegaTOF, Large Molecule Biotherapeutics, and Protein Interactions Analysis on a Benchtop platform. Before we start, a couple of notes for everyone's viewing experience. The webinar console has a variety of items to help enhance your experience and interaction with us. So in the screen, you'll see the following. On the right are the survey questions that you may fill out anytime during or after the presentation. On top are the speaker's bio. You may expand the items here to learn more about us. In the center is the area for presentation slides, and below these slides are the resources. In here, you'll find links to application notes relevant to the topic. On the top left-hand side is the widget for question and answers. Please submit your questions during the presentation through this widget, and we will answer them either during the Q&A session at the end of the presentation or after the webinar concludes. Finally, just below is a demo request form. If you are interested in seeing the instrument personally or have samples to submit for evaluation. All right. Now that you are a little bit more familiar with the viewing platform, we're pretty much ready to start. Again, if you are joining us, I'm Nazim Boutagu, your moderator. Today, we will be discussing large molecule biotherapeutics and protein interaction analysis using a unique benchtop MALDI. We ask that you submit your questions through the widget, and we will do our best to answer them at the end of the webinar during the Q&A session. We will be starting off with a presentation from Dr. Ryan Wenzel. And before we pass it over to him, I would like to give you a brief overview of his background. So Dr. Ryan Wenzel received his PhD from Professor Kim Prather while studying aerosol mass spectrometry at UC Riverside and UC San Diego. He then completed a postdoc at the ETH in Zurich under Professor Renato Zanobi, researching large molecules mass spec analysis using cryogenic detection. From work at the ETH, together with Alexei Nazarov, they founded Covalx in Switzerland in 2005. In 2008, Ryan moved to the Boston area and established the U.S. subsidiary of Covalx, where he is currently located. Throughout his position, he has been involved with mass spec instrument design and ion detection. Currently, Ryan is the Director of North American Operations at Covalx Instruments, located in Saugus, Massachusetts. He manages the new products from Covalx, including the development of the OmegaTOF Benchtop MALDI instrument in collaboration with Shimadzu. Okay, without further ado, I will hand it over to Ryan.
Thank you, Nazim. I'm Ryan Wenzel with Covalx, and I'm excited to share with you today's presentation on the latest collaboration between Shimadzu and Covalx for high-mass ion detection using a benchtop MALDI, which we're calling the OmegaTOF. Brief overview of what I'll be discussing with you today. Let's start with some background on mass spectrometry, MALDI in particular, and specifically high-mass ion detection. Then move into some of the specifics of the OmegaTOF instrument before showing some applications, some example data, and key applications we've developed, and then finally wrapping everything up with a summary. As I said, first I'll start with some basics of mass spectrometry, specifically MALDI and high-mass MALDI.
Okay, so the OmegaTOF utilizes MALDI time-of-flight mass spectrometry. So MALDI, of course, stands for Matrix-Assisted Laser Desorption Ionization. And what we're going to do here is we're going to take the analyte, the sample, typically a protein, and we're going to mix it with the matrix molecules. The matrix for proteins that we're going to be using is cinnapidic acid, and we're going to spot that on a MALDI target. The MALDI target is a metal plate, typically cinnapidic acid, where the sample and the matrix can co-crystallize onto the target before we introduce it into the vacuum. So now that we've got this mixture of analyte matrix into the vacuum, co-crystallized on the target, we're going to apply a laser light. It's a laser pulse. It's a pulsed laser. It can be used to desorb the molecules, so it's going to heat up the molecules, desorbing them, that's the D in MALDI, and then ionizing those molecules. That's, of course, the I in MALDI. So the matrix is going to both absorb laser light and then transfer ions into the sample, the analyte, which can then be later separated down the time-of-flight analyzer and detected by the detector. Okay, the mass analyzer or ion separator that the OmegaTOF uses is, of course, a time-of-flight or TOF mass analyzer. And the way this works is the ions are going to fly down the time of flight and be detected upon impact at a detector. So, as discussed in the last slide, we'll create the ions with by mixing them with a matrix, and then we'll use a pulse of laser light, and the ions will fly down the time of flight being separated. Of course, lighter mass ions, lower mass ions, will move faster down the flight tube and impact the detector first, and heavier, slower ions will move later down the flight tube and impact the detector at a later point. So this will therefore separate the ions, each laser pulse being the start of the experiment, and the impact on the detector being the end of the detection. One critical equation for this is the Ke equals 1/2 mv squared. Kinetic energy equals 1/2 mv squared, which is critical for both the separation as well as the detection once impacting the detector, as we'll get to in just a moment. Now, there are a couple issues with detection of ions with most standard detections. One of the traditional detectors is an MCP or microchannel plate detector, but this can be found with most standard detectors. The two major issues that we'll be addressing are both sensitivity and saturation. So sensitivity is that as ions get to higher and higher masses, detection efficiency goes down. This typically becomes noticeable in a time-of-flight experiment in the tens of kilodalton range. Becomes quite significant, and detection becomes limited most of the time with instruments in the hundreds of kilodalton range. And detection of ions in the megadalton range is typically not possible, aside from some of the high mass detection that we'll be featuring in these instruments. The second issue is the issue of saturation. This is an issue with complex samples where early arriving ions will hit the detector first, making it more difficult for later arriving ions. So this is an issue we call saturation, and both of these issues will be addressed to some degree with the high mass detection featured in the OmegaTOF. Now, to go into a little bit more depth on these issues with standard detectors, both the issues of sensitivity and saturation, we'll look a little bit at how the standard detector works and some of the basics and concerns behind this. So here's an example of an MCP or microchannel plate detector. The same fundamentals work with most standard detectors, even if they're not microchannel plate detectors. And that's that incoming ions are going to impact the surface, creating secondary electrons. Those are going to be amplified and then detected directly as current, as output electrons. Here we see on the left a crosscut of an MCP or microchannel plate detector, which is many tubes, many microchannels attached together to create this plate. And on the bottom right, we see a front view of one of these microchannel plate detectors, a cartoon of it. And the way these work, again, are the incoming ions are going to impact one of the tubes of the detector, and they're going to eject secondary electrons upon impact. And those electrons are then going to cascade through the detector, creating more and more electrons as they bounce off of the surfaces. And this cascade or amplification of electrons can then be measured as output voltage on the back end of the detector. One of the main concerns with this is that as the ions get larger, so if we assume the basic time-of-flight equation, Ke equals 1/2 mv squared, as we discussed earlier, if we've got fixed source voltages, 15, 20 kV source voltages, and we've got singly charged ions, we've got the kinetic energy is fixed. As the mass goes up, as the ions get to be larger and larger ions, the velocity is going to go down. They're going to be moving more and more slowly. And the issue that this can cause for sensitivity is that this ejection of secondary electrons, the efficiency of this begins to go down with slower and slower ions, so detection efficiency becomes an issue. This issue with detection efficiency or sensitivity becomes noticeable in the tens of kilodaltons for standard time-of-flight experiments, and becomes a more significant issue in the hundreds of kilodaltons, which is often where the limit of detection for these is, and makes it typically not possible to detect ions in the megadalton range, which is specifically what the OmegaTOF is designed to do. So that's the first issue with sensitivity with most of these standard detectors. The second issue, which we mentioned earlier, is this issue of saturation. And saturation is where early arriving ions are going to hit some of the tubes of the MCP or the detector. They're going to cascade through, discharging that tube of the MCP, therefore making it turned off for the rest of that time-of-flight experiment. It will charge back up for later laser shots, but on that time-of-flight experiment, it will be discharged, making the detector less sensitive due to this saturation effect. So if we look on the bottom right here, the detector, what's going to happen is as the early arriving ions hit the detector, it's going to turn off some of these channels, and then more and more channels will be turned off, again making it, saturating it for detection of later arriving larger mass ions. So even if it is possible with a pure sample to measure something at higher masses, when you work with a real sample that has multiple ions arriving at earlier masses, detection can be more and more difficult due to this saturation effect. In the OmegaTOF instruments, we utilize one of the Covalx high mass detectors, which are specifically designed to correct for as much as possible these issues of sensitivity and saturation, which we've been discussing. The main difference here is that the incoming ions from the time-of-flight experiment, upon impacting, don't directly create electrons, which are then measured, but instead undergo an ion-to-ion conversion, creating secondary ions upon impact of an ion conversion dynode. So there's a special a special ion conversion dynode at the front of this detector that upon impact of these larger, lower velocity ions can more efficiently kick off secondary ions. However, these secondary ions are not moving very rapidly to kick off, to create electrons, so we have to re-accelerate those. So there's a secondary acceleration field inside this high mass detector of actually 20 kV inside the detector over a very short distance. So a high voltage over a short distance that can re-accelerate these secondary ions before impacting them into an electron multiplier type detector that can create the ion to electron conversion and then amplify those, similar to what we discussed before. So again, the two issues of sensitivity. First, the ion-to-ion conversion is much more sensitive for these larger, low-mass ions. And second, the issue of saturation. So not shown here, but this style of electron multiplier, we actually add extra capacitance onto the back of it, some of the later plates of the detector, so that it can handle a very high current, so that even early arriving ions that provide a very strong signal will not saturate the detector and make it very sensitive for complex samples. So this will take advantage to improve both sensitivity and saturation. Okay, so we've talked about MALDI, about the ionization, about the separation, about the detection, and the ability to measure these proteins. And MALDI is known as a soft ionization, one of the softer ionization processes. This is one of the reasons it won the Nobel Prize. However, while it does work for measuring proteins in most cases, it's not efficient or capable of measuring protein complexes or protein interactions directly in the mass spectrometer. What will happen with these non-covalent complexes is that they will dissociate either during the sample preparation with the matrix or during the vacuum stage or ionization stage. The complexes will be dissociated. And so to get around this, what we want to do at these experiments with the OmegaTOF is look at these protein complexes intact. And so again, what would happen is if you had a protein complex here, protein complex AB, say an antibody antigen complex, when you ionize it with the laser beam, what you're going to detect is protein A and protein B separately. This is great, but if you want to see the complex, see the stoichiometry, measure that the complex is interacting, what we'll need to do is hold that complex together during the experiment. And so in order to do that, what we'll use is chemical stabilization, cross-linking chemistry, to turn that non-covalent complex into a cross-linked, stable covalent complex. Now when we ionize it with the laser, what we can instead detect is not just A and B separately, but the intact complex and in fact the stoichiometry. Of course, this is going to be at a higher mass, therefore the OmegaTOF detection comes into play here. In order to do this cross-linking chemistry, what we utilize is homobifunctional amine reactive cross-linkers. Typically, we'll utilize cross-linkers of different lengths, from 6 to 18 angstroms, a mixture of different lengths of cross-linkers depending on the complex that we're looking at. These are non-cleavable, not reversible cross-linkers that react to 6 different residues on the protein's structure, but provide an efficient, fast method for cross-linking and stabilizing our complexes. Covalx does sell these as kits. We have different kits depending on the size of the complex that you're looking at, the K50 to K200. Most of what we'll be utilizing today is the K200 cross-linking chemistry, again, to hold together covalent complexes up to the megadalton or even larger mass range. And we'll show you some examples of those in just a moment. So that was some basics on mass spec and some of the fundamentals and limitations of mass spec. Now I'm going to switch gears and talk a little bit about the OmegaTOF itself, about the high mass benchtop instrument that we're promoting with Shimadzu. So here we see the Shimadzu OmegaTOF. This is powered with the Covalx detection system. This is the first benchtop linear high mass MALDI instrument to feature the Covalx high mass detection. So it's built on the platform and fundamentals of Shimadzu's benchtop platform that had a compact design, fast analysis, the latest softwares, some special maintenance capabilities, which I'll get into as well. So a very advanced instrument, all in a benchtop platform. This is designed specifically for measuring larger molecules, so again, things in the hundreds of kilodaltons, tens of kilodaltons, hundreds of kilodaltons, into the megadalton range. Here we see examples above into the megadalton range, and I'll show other examples. So this, again, utilizes Shimadzu's latest benchtop platform with the fast interlocking stages, all the latest turbo pumps and lasers, as well as the implementation of our latest 5th generation, the Covalx's latest 5th generation detector systems and power supplies, all controlled in the standard user interface. Again, this is a benchtop instrument, so it's a very small footprint, less than half a meter wide, by just over a meter, 1.2, 1.18 meters high. So fits nicely onto a desk-sized platform with the PC next to it or in another room for ease of use. One of the benefits of this latest benchtop platform is very fast sample introduction, about 1.5 minutes for sample introduction, so you can rapidly switch between target plates. Each target plate can handle multiple samples, so a very high throughput process. The laser utilized here is a 200 hertz solid state laser, so long lasting, very stable, and a good rep rate as well. The software package built into the OmegaTOF instrument utilizes Shimadzu's CFR21 Part 11 compliant software. So all the security features that are needed, user management, audit trails, necessary to follow CFR21 Part 11 compliance are included in this platform. The source utilized also takes advantage of the latest technology from Shimadzu. It's got all the latest ion optics as well as their TrueClean UV source cleaning option. Additionally, if necessary, the source can very easily be removed for cleaning. It has the longest lasting MALDI laser, as I mentioned, that solid state laser, so a very robust, long lasting laser. Additionally, not going to get into this too much today, but it's nearly an on-axis firing, which provides a higher efficiency and sensitivity, especially for larger mass ions. So the laser's coming in nearly orthogonal to the target plate, so the ions are ejected straight off the target plate, allowing again for higher efficiency for these larger mass ions. So in summary, the OmegaTOF is the only benchtop option featuring the high mass capability. It provides access to all the Covalx applications from protein-protein interactions, specifically monoclonal antibodies. I'll show you some examples with aggregation and polymers as well. It's priced competitively at an entry-level pricing. Very ease of use, walk-up friendly type of instrument. So not a lot of training or advanced knowledge necessary to operate this instrument. Very fast sample introduction and analysis time. Being that it's MALDI, very low cost per sample, as well as ownership costs, so not a lot of consumables or parts that will wear out. And as I mentioned, the 21 CFR Part 11 compliance. So in all, a very robust instrument with very unique features, specifically, again, focused on the high mass applications. How to get in those applications, I'll show you some examples of things that can and have been done on this OmegaTOF high mass MALDI benchtop platform. So the examples I'm going to show you start with just some simple, straightforward, but very novel large molecules that we'll look at intact, IgM, KLH, then move into some protein antibody interactions, some PEGylated proteins, aggregates, and then finally end with some polymers as well. So for large molecules, this is one of our kind of flagship examples that we show is IgM. So immunoglobulin M is the larger of the IgM family. It can be found either as a pentamer or a hexamer. Here shown as a pentamer at 970 kilodaltons. When created as a hexameric molecule, it'll be just over a megadalton at 1.2. This technique, this instrument can be used to measure it directly, very rapidly. One thing that is unique and interesting when you get to these very large molecules, what we see here is the primary ion at about 970 kilodaltons, but we also see double charged ions at 486, triply charged at 325, and quadruply charged at 244 kilodaltons. So we see not just the singly charged ion, which is predominant in MALDI, but when you get to really large molecules like this, you can get some double, triple, or even quadruple charged as stated. But we are able to detect without deconvolution directly from the mass spec. This is what the data looks like coming out of it. We can see the megadalton massed ion. We can do this also for deglycosylated versus native, from sec fractions, lots of different possibilities. Example of both pentameric and hexameric samples being measured in the high mass. So we can see for pentameric samples, again, they're below a megadalton, in this case around 940 kilodaltons, and the hexameric sample at over a megadalton, as I mentioned. So we can see those differences there directly. Okay, here we have just another example of a very large molecule being detected with the OmegaTOF platform. So this is a KLH, or keyhole limpet hemocyanide. This is nearly half a megadalton, 467 kilodaltons as a singly charged ion. Again, as we showed with the IgM, one thing unique to these very large molecules is that we measure them not just as a singly charged, although we do see that in this case at 467 kilodaltons, but we do see some double, triple, or quadruple charged, in this case at 230, 178, or 120 kilodaltons. But the largest ion and very direct detection is at 467 kilodaltons. This is measured at a very low concentration, so measured at 250 nanomole per microliter. This is a very common carrier protein found in antibody production, a glycosylated protein. There is an app note available from Shimadzu on this with the OmegaTOF. Okay, I gave a couple examples of large molecules being detected directly, proteins, but mentioned looking at protein-protein interactions. And here's an example of just that, an immunocomplex, a simple antibody-antigen complex that we're going to measure in the mass spectrometer. As I mentioned, when you put it in the mass spec, the complex dissociates, and that's what we see in the left in the blue spectra. Here we have a basic interaction between an antibody, 6H4, and its antigen, BPRP, bovine prion protein. In the left, lower left control spectra in blue, what we see is the antibody at 149 kilodaltons, and the antigen, in this case, at 24.5 kilodaltons. As mentioned, these two should be forming a complex, but when you conduct the MALDI experiment, the complex dissociates. So we use cross-linking chemistry, and that's what's shown on the right. It's after using cross-linking chemistry and measuring the complex directly. What we see is not just the antibody at 149 kilodaltons, but the 1-to-1, and more specifically the 2-to-1 complex, in this case at about 200 kilodaltons at 198 kilodaltons, with 2 of the antigens binding to 1 of the antibodies. And the antibody has 2 binding sites, so we see that 2-to-1 stoichiometry quite selectively and specifically. So this is a direct measurement of that complex, very straightforward, as well as using the OmegaTOF for the high mass capabilities up to a couple hundred kilodaltons. An example of another immunocomplex shown here, a little bit bigger antigen of 107 kilodaltons. The antibody again is about 143 kilodaltons. These two should be forming a complex, but in the control experiment shown on the left in blue, we see the antibody and the antigen separately. Down below, in the after cross-linking, we see the antigen depleting and a new peak forming from 1 antigen and 1 antibody. So this is actually a bispecific antibody, so there's only 1 binding site on it. So we can detect that quite selectively, in this case out at 248 kilodaltons. This as well was shown in an app note with the OmegaTOF data available from Shimadzu and here on the webinar. And we've done lots of antibody-antigen complexes. On the top left, we see a larger antigen of 135 kilodaltons with the antibody, again, a full MAB at 150, forming a 1-to-1 and more specifically a 2-to-1 complex, in this case out at 418 kilodaltons. Again, with the high mass capability, it is a direct measurement of these proteins out at 400 kilodaltons. On the top right, we see BSA forming a 2-to-1 complex, in this case out at 282 kilodaltons. Something smaller on the bottom left with myoglobin at 16, 17 kilodaltons. And finally, a small antigen of a beta amyloid fragment against a full MAB, showing the 2-to-1 complex at just 161, so a 5 kilodalton beta amyloid interacting. Here we see another immunocomplex, but in this case we're actually trying to see if the complex is binding against antigen X or antigen Y. So we've got 2 different antigens, one of about 24.5 and one of about 26 kilodaltons. These 2 are in the mixture with an antibody. This is an antibody that we want to see if it can bind X or Y. And so what we see is after cross-linking, it is actually binding antigen Y. So we see it being detected in this case again at about 200 kilodaltons. And antigen X at 24.5 is still present in the mixture. So we can use it to selectively bind and detect which target it's binding against. Next, a very relevant example to a lot of today's research is the target antigen SARS-CoV-2. This target is 88 kilodaltons. Here we're binding again full MAB, a full monoclonal antibody of 149 kilodaltons. And again, in the control spectra, we see the antigen at 88 and the antibody, in this case, at 148.9 kilodaltons. These two should be interacting after cross-linking on the lower right. What we are able to detect is the depletion of the antibody and a 2-to-1 stoichiometry. So this is a standard MAB with 2 binding sites. So we are able to detect 2 antigens, 2 of the SARS-CoV-2 antigens binding directly to the antibody, in this case out at 343 kilodaltons. Again, this example, there is a note on the OmegaTOF available here on the webinar or from Shimadzu. This is an example of a little bit more complex antibody. It's actually a DVD, or a divalent antibody. This antibody is actually engineered to bind 2 different antigens onto each arm of the Fab at the same time. And that's what we were wanting to see if we could detect here. So in the control experiment, we see the 2 different antigens, A and B. I believe they were 18 and 65 kilodaltons, and the monoclonal antibody, in this case at about 200 kilodaltons. A little bit bigger because it was engineered to have those longer Fabs to bind both targets at the same time. And what we were wanting to see if it's possible is to detect that full complex, if that's actually able to form that stoichiometry. And on the lower spectra in red, what we see is the primary peak at 383 kilodaltons. Again, with the high mass detection, a direct measurement, it's several hundred kilodaltons. And this corresponds to the 2-to-1 stoichiometry as the predominant peak, as was hoped to be formed from this sample. Additionally, we can see some of the less intense peaks from the 1-to-1 stoichiometries, 2-to-1, other 2-to-1 are present there, but the predominant peak is, as hoped, the 2-to-1 stoichiometry quite clearly. It is common during antibody manufacturing to generate multiple monoclonals against a given target, and then group or bin those down to antibodies that have similar binding to the target. So they bind onto similar groups or epitopes on the target antigen. One of the ways to do this binning or grouping experiment, or as we call here sandwich assay, is to take 2 different antibodies, 2 different monoclonals, and see if they're able to bind onto the target at the same time. The way we do that is in this example here. We've got the blue and the pink antibody, and they're both able to bind onto the target antigen, but not at the same time, the blue and the pink. So what we see in the spectra on the top right is we see an unbound antibody as a small peak, a 1-to-1, and then a larger 2-to-1 peak. We can't discern between the blue and the pink. They're similar masses, but we can see them both binding 2-to-1. In the lower case, though, we have the blue and the black antibody. These are able to bind onto the same time onto the target antigen. And so what we see here is a depletion of the 2-to-1 peak from the top spectra going down to the lower spectra. So the 2-to-1 peak becomes lower in intensity, and a new peak is forming with 1 antibody, 2 antigens, and a second antibody, in this case out at 356 kilodaltons. So again, using the cross-linking chemistry to stabilize these complexes and the high mass detection to measure them. This as well, we have an app note with data on the OmegaTOF. Here's an example of this shown directly here. Again, in this case, we're showing the blue and the orange antibody binding onto their target antigen, in this case out at 383 kilodaltons. So we can confirm that these are different epitopes or binding regions between these 2 MABs. Here we have an example of a modified or PEGylated protein. PEGylation is where we take polyethylene glycol, or our clients take polyethylene glycol, and conjugate it to the surface of a protein. This can have several benefits for the protein itself, increasing half-life, increasing stability, having some benefits with extending patent possibilities, hydrophobicity, reactivity. So there's several reasons a client may want to PEGylate a protein. But upon doing that, they then want to characterize the degree of PEGylation. And using the OmegaTOF, here we see an example of a PEGylated protein being measured. So we've got three different examples of it. First, we have the unmodified protein with no PEGylation present. It's about a 60 kilodalton protein, so we can measure that unmodified. In the middle example, we have it with a lower degree of PEGylation. This is a 21 kilodalton PEG, so we can see that begin to become PEGylated. And then in the final example at the bottom, a higher degree of PEGylation. And so here we see peaks out beyond 100 kilodaltons. It's centered around 2 PEGs, which is 108 kilodaltons. And while this doesn't provide a quantitative measurement of the degree of PEGylation, it does provide a much more accurate measurement due to both the sensitivity and saturation effects that our detector helps to solve. Aggregation is an issue that many of our clients experience when developing biotherapeutic protein drugs. This can be not just for antibodies, although it's definitely an issue for antibodies, and that's what's shown in this example. But for any protein biotherapeutic, aggregation can be a concern. Historically, it's something that's measured later in the development stage, but it is something that's been pushed sooner and sooner to select out for developability. And so having a more rapid and direct measurement of characterization is crucial. On the top here, what we see is an SEC, or size exclusion chromatography trace, for a stressed antibody. So this antibody has been stressed to induce a significant degree of aggregation, and then those different fractions were collected and ran by high-mass MALDI. Early arriving fractions show higher degree of aggregates, tetramers, trimers. As you go later into the time frames, you see the trimers and dimers, and then finally the primary peak of only monomer. And I'll show you in a little more detail some of these spectra as well. Important to note, typically this would be measured either just with a UV trace based off the time, or potentially doing something like SEC-MALS, so a multi-angle light scattering experiment. Those multi-angle light scattering experiments are going to take an average of what's measured underneath for a given fraction underneath a given sample, and not provide specific insight into the type of aggregates or oligomers that are being detected. And with the high mass MALDI, with the OmegaTOF, we can specifically see those types of aggregation. So as you step through the SECs, first in the earlier traces, again, we see the higher mass aggregates. So the predominant peak here being a dimer at about 300 kilodaltons, trimeric species at 450, tetrameric, pentameric, all the way even hexameric species can be seen here out at over 700 kilodaltons. Again, this is a direct measurement from the SEC fractions, so there's no deconvolution necessary. This is what it looks like coming directly out of the instrument, out of the MALDI TOF. As we step later, SEC fractions, we can see again lower mass ions. Here we see predominantly the trimeric species being separated from the SEC. We can see some double charge from the trimer, which is interesting again for these very large ions. We can see double charge, so the trimer being at about 450 kilodaltons, the double charge of that being at 225 approximately kilodaltons. As we step to later times in the SEC, we can see specifically the dimer, quite a clean dimer sample as well. I mean, obviously some monomer that could also include some double charged dimer, but very clean dimer separation from the SEC here at 6.9 minutes. And at the latest time point from the monomer, we can see the monomeric peak again at 150, 148 kilodaltons, being the monomer peak coming out of the SEC at 8.1 minutes. Now to shift gears a little bit, a lot of the examples I've shown you so far have been on biotherapeutics, mostly antibodies and elements around antibody development and characterization. But the OmegaTOF doesn't have to be used just for antibodies. It can be used for any type of large molecule protein or other types of protein-protein interaction, protein complexes. Here's an example of a simple three-component complex, an AMP kinase, composed of an alpha, beta, and gamma subunit. In the top trace, in the black, we see the alpha, beta, and gamma in the black spectra, all at different masses, 30 kilodaltons, 37 kilodaltons, and 65 kilodaltons. Again, these complexes will dissociate, these non-covalent complexes will dissociate when you put them in the mass spectrometer, and that's what we see in the first black trace from the control. In the red on the top, or in the black after we subtract out the differences, what we see is one primary peak from the 1-to-1-to-1 complex due to the alpha, beta, gamma 1-to-1-to-1 stoichiometry, which directly aligns with multiple other measurements that were conducted on this sample, non-mass spec measurements, to confirm the stoichiometry that was believed to be occurring. Interactions, and again, there's many of these that can be shown as examples, but just a few for today's webinar. Here is an HIV integrase with LEDGF. These two were believed to be forming a complex, but again, the stoichiometry was not known. Again, it's a non-covalent complex, so when we measure it directly in the mass spectrometer, what we see are the subunits of the complex being measured. So we see the integrase at about 33 kilodaltons and the LEDGF at about 63 kilodaltons. However, after cross-linking and stabilizing this complex, they were able to measure specifically a 4-to-2 stoichiometry, again, which is in agreement with other measurements as shown in the publication. So again, a very direct measurement of the stoichiometry for a protein-protein interaction. Of note, these researchers then took it a step further and added a third component where they did a stoichiometry of an even larger complex. And so you can build these complexes and look at their stoichiometry directly quite rapidly in these types of instruments. Membrane proteins is something that is obviously of interest, and we've seen a great degree of interest in analyzing these types of samples. The main concern that we often address with membrane proteins is solubility. So if the samples can be solubilized, then it is potentially possible that we can measure them and measure these complexes. This is an example from a few years back of a publication from a membrane complex, which was solubilized in a couple different detergents, and then able to actually be measured as a control, where we see the subunits. And then after cross-linking, we can actually stabilize and measure the complex directly of this membrane complex. Finally, one last additional protein-protein interaction, and again, there's many of these. Feel free to reach out to Shimadzu or myself if you're looking for additional examples. But a Saga hat subcomplex, this was a more advanced protein-protein interaction network, and using the high-mass MALDI, they were able to characterize stoichiometry and the interaction of some of the different subcomplexes of this network and submit to this publication. Lastly, before wrapping up the webinar, I just wanted to mention a couple quick examples on polymers. First thing to note is ionization of polymers can be different than ionization of proteins, biotherapeutics, antibodies, as I've been discussing before. So selecting the right matrix and ionization conditions can be a little bit more of a challenge than what's often seen with proteins. That being said, it is possible, and I'll show you just a couple of examples. I already showed you PEG when it was conjugated to proteins, but here's some examples of polymers directly. First is a dendrimeric polymer that was measured with high mass detection. So there were several different dendrimers that were generated, different generations. You can see them growing from the top here, G1, G2, G3 as you go through. And then these were measured directly by high mass. We did SEC separation to show that we could separate out the different generations, G3, G4, and G5, and then measured them in the high mass instruments. So you can see for the G4, we see peaks centered of these dendrimers around 65 kilodaltons for the monomer, but it's actually forming a tetramer out above 200 kilodaltons, with peaks extending beyond 500 or 600 kilodaltons. For other dilutions, we were able to get cleaner separation of the tetrameric peaks, as shown in spectra B. Again, with the tetrameric peak being the predominant peak at just over 260 kilodaltons. And then for the G5, even larger, above a half a megadalton for the tetramer. The monomer of this was 134. These dendrimers provide very clean peaks that could be isolated for the different aggregates. And finally, one last example of another polymer species. This is a dendritic porphyrin fullerene hybrid. This is some work that was actually done by some users in Austria with a high mass detection. And on the top, we see a subunit of about 6,000 or 7,000 daltons, with species being measured up almost to 100 kilodaltons. And on the lower case, species centered around 20 and 30 kilodaltons, again extending to the 100 kilodalton range. One thing, again, as I mentioned with polymers, that is critical is the selection of matrix. And so here they actually used a different matrix, CMBT, as highlighted. In summary, we started first with a basic discussion of MALDI, some of the principles of MALDI time of flight analysis, some of the limitations for MALDI detection of very large molecules, specifically sensitivity and saturation were discussed. And then we went into the introduction of the OmegaTOF, the first benchtop instrument specifically designed for high-mass MALDI applications, featuring the Covalx detector, and discussed how we can address those issues with sensitivity and saturation in the latest benchtop technology. Then moved into some different applications featuring this technology, specifically some large molecule analysis, examples of IgM, immunoglobulin M, KLH, some other big proteins. Then moved into some antibody antigen interactions, showed different stoichiometries, some small antigens of a few kilodaltons up to several hundred kilodalton antigens into the half megadalton range, different antibodies from simple MABs to bispecifics with 1-to-1 stoichiometries, or divalent antibodies with 2 to 2-to-1 stoichiometries. Some examples of covalently PEGylated proteins were then shown, showing the distribution of the PEGylation across the proteins. Protein aggregation was shown next with some examples from size exclusion fractions, looking at different aggregates from several hundred kilodaltons for antibodies up to 600 or 750 kilodaltons down to the monomeric fraction. Next, we moved away from antibodies into some protein-protein interactions, looking at those intact. Different stoichiometries were analyzed with an example of a membrane protein complex given at the end of that. And finally, wrapped it up with a couple of examples of some polymers, something a little bit different, but some larger polymers being analyzed with the high mass detection. So with that, I think we'll bring back Nazim and address any questions that may be asked, and look forward to addressing them.
Thank you, Ryan, for such an insightful presentation. I would like also to thank our audience for attending and sending questions. I think we have time for a couple of questions, and we will answer some of them. Ryan, are you ready?
I'm ready. Let's go.
All right. So let me look through it. All right, first question, and this, yeah, I think you can answer this one. Can you perform drug-antibody ratio experiments?
Oh, yeah. So DAR ratios, that is something we've done with the high mass detection. It does depend on a couple things. One, the size of the drug or the molecule that's being covalently bound. If we're dealing with a smaller molecule, it may be difficult to resolve it on the larger protein. If there's multiple or several drugs or molecules covalently linked, then seeing that distribution, seeing the peak shift is something we've had success with. Additionally, if the moiety that's being covalently bound, something like is something larger, like the example I showed with the PEG in the slides here, there we could definitely resolve each individual unit, subunit, and give a much more accurate distribution. One thing that is important with the OmegaTOF is the lower saturation that we discussed is going to provide, while it's not quantitative, it will provide a much more accurate distribution of the actual ratio, drug ratio, DAR ratio in the samples. So that is possible depending on the sample.
Nice and thorough. Thank you, Ryan. Second question. Do you have access to lower molecular weight species analysis? How does it compare to the regular detector? And third part of the question, what is the resolution of this instrument?
Okay, I'll take that one. Yes, the OmegaTOF can measure lower molecular weight species. It can measure into hundreds of Daltons or a few thousand kilodaltons if needed. However, I will say if that's your focus of your research, you're probably going to be better off with an instrument that's more dedicated towards that, that doesn't have the high mass detector like the 8020 or 8030 from Shimadzu. Reason being is going to be the speed of the detectors in those and therefore the resolution when you're dealing with lower mass ions. So for things below or above 10,000 in the 50,000 range or higher, the speed of the detector isn't an issue and it's not going to be as noticeable. And that's really why the OmegaTOF focuses on the large molecules, but it can measure smaller ions. As far as resolution, if you look at something like an antibody at 150,000 Daltons, typically we see a resolving power of about 100, which is going to give us the ability to fully baseline resolve about 1,000 Daltons shift up at 150,000. So resolving power of about 100 in that range. So
very good, very good. Thank you again, Ryan. Moving on, question number 3. How many samples can you analyze on a target plate? I think I can take this one, Ryan.
Yeah.
So we have a smaller source on the MALDI 8020 and 8030, and it accommodates like a plate that has the size of a microscope slide. So basically, we have 48 sample spots on there, and on top of that, you can add 3 calibration spots. These are located between, the sample spots. So for 52 spots, 3 of them usually that we recommend for calibration. Moving on, question number 4. I think you may want to take this one, Ryan. Can you use this to measure binding kinetics of the interaction?
Yeah, so the short answer is no. Looking at binding kinetics, while that does align with a lot of what we do with protein complexes, protein interactions, looking at binding kinetics is not something that we can do with the OmegaTOF. That's something we'd really recommend more with something like SPR or BLI type instruments to get the binding kinetics. But one thing of note with those instruments is that they often need to assume the stoichiometry, the ratio of the interaction. And that is something that the OmegaTOF and some of the examples I showed can be useful for. So while it doesn't measure binding kinetics, it is somewhat orthogonal and does contribute in some ways to those measurements. So good question.
Okay, also great answer. Thank you, Ryan. Fifth question, and because of our time, this might be actually the last one we will take.
Okay.
Let me look. All right. How long does the laser last, and what are the consumables needed to operate the OmegaTOF? I think, yeah, Ryan, I'll take this one.
Yeah.
Yes. So our laser is spec'd for 2 billion shots, which basically on the paper makes it actually one of the longest lasting lasers available on the market. And regarding the consumables, I think when it's more related to MALDI than I think OmegaTOF. Although OmegaTOF and Covalx sells their own set of consumables that were probably discussed earlier. But in general, I mean, you'll need our plates, and these are provided with the purchase of the instrument. And we have two types of plates. We have polymeric ones. We have also stainless steel. You'll need solvents, so regular MS solvent, water, acetonitrile, methanol, and for MALDI, of course, we like to use TFA. And pipettes are actually very specific because we pipette very, very small volume. Basically, we spot a half microliter to a microliter on each sample spot. So you need to have specific pipettes that can handle such low volumes. So P2s or P10s are probably are very much required for that, and also the pipette tips that come with it. So in terms of matrices, and specifically for OmegaTOF, because we're looking at protein analysis and higher molecular weight protein analysis, it's going to be synapenic acid. That's going to be really your go-to, right,
Yep.
Ryan? I think you guys use a lot of variety.
That's 100% correct.
Matrices.
That's what we do all the proteins with, the synapenic acid, yes.
Yes. And of course, you did mention that there's also very versatility in terms of samples we can analyze. So when it comes to polymers, it opens up the door for like a whole plethora of matrices and a lot of options there. But, we're not going to go through them. But yes, you're right. I mean, if you start doing polymers, it might call for different matrices. But we will be happy actually to answer those questions if anybody is really interested in diving a little bit more precisely into that type of analysis. So contact us and we will answer that part. So that was question number 5, and that's pretty much it for the presentation and the questions also, due to the time that is allocated for this webinar. So Ryan, thank you again for making yourself available and presenting. This was truly insightful, and I truly believe we also have a game changer here for higher molecular weight analysis. Before we move it, do you have any comment?
No, thank you, Nazim, for coordinating this. I'm also excited for the new technology with the Benchtop. My email's here. Feel free, if anybody has questions, to reach out to me directly, or reach out to somebody at Shimadzu, and they can. We'd be happy to address any questions. Thank everybody for listening.
So once again, thank you all for attending. We will send you an email with a link to view a recorded version of this presentation anytime. We hope to have you back in webinars we organize in the future. Have a great day.